Morning gang, we are going to do some work on T cells today. So there's B cells and there's T cells. I'm going to split this up into two. So today when we're talking about immunity, the type of immunity we're going to talk about is what's called cell mediated immunity, which is essentially things called T lymphocytes. Okay, so sometimes they're called T cells, preferably called the lymphocytes. Cyte means cell, lymph, they hang out in the lymph. Okay, T lymphocytes are made in the bone marrow, they mature in the thymus, and they're part of the immune response. If I just do this section on T cells, then when I build on it next with B cells, you can stick the whole lot together. Okay, so cell-mediated immunity, what does that actually mean? It means that T lymphocytes only respond to antigens which are on cells. Okay, so for example, a phagocyte which has engulfed, so we talked about last lesson, we talked about a phagocyte engulfing a pathogen. When it's done that, what it does is it presents the antigen on its surface. So any phagocyte, any phagocyte which has engulfed a pathogen will display the antigen on its surface, and T cells can deal with that. The other thing T cells can respond to is host cells, so as in body cells, which have been infected, and what they basically do, those cells which get infected, especially ones infected with viruses, will essentially rip off the antigen and display it on their surface, essentially going, help, help, I've been infected, so that your T lymphocytes actually know what they're dealing with. So T lymphocytes respond to only cells presenting antigens. They don't respond to the pathogen itself. Okay, now the first stage of things that happen, I'm going to choose a different colour. The first stage with your T lymphocytes is basically selection. So they need to select the correct T lymphocyte. So all T lymphocytes have receptors on them, and I'm not going to draw a ton of them, but I'll just draw two. But you can see from my diagram that this T lymphocyte has a receptor that is complementary to the antigen, whereas this one doesn't. Okay, so the first stage is selection of your T lymphocyte, the one which has got the complementary receptor. The next thing that happens, so these then will bind together, and what that does is it causes those T lymphocytes that have been selected to be cloned. So the next thing is there is cloning. That cloning happens by mitosis. So this T lymphocyte now becomes a shed load of complementary T lymphocytes, because one just ain't gonna be good enough. So you end up with a bunch of T lymphocytes that are complementary to the antigen on the cell that is presenting it. Sorry, I did not move my book. It's not very helpful, is it? So we've got an antigen presented on a phagocyte or a host cell. The complementary receptor on the right T cell has been selected for. That's then cloned by mitosis, so we have a lot of them. Then the next stage is differentiation. So what happens is the T cells can turn into different types dependent on their job, and we will then look through what their job might be. So the first type would be a TC, sometimes called TK cell. This stands for cytotoxic, and these are the ones which actually can go and destroy host cells. So let's imagine that this has got, well, let's go for COVID-19, shall we, inside it, and it's displayed the antigen on its cell surface membrane going, I've been infected with COVID-19, and your T lymphocytes will clone and differentiate and produce TC cells, which will go and destroy that host cell and be like, sorry mate, but you're infected, we need to kill you. And that's how the viruses get destroyed. Okay. The way they do this is they have an enzyme called perforin, which, as the name suggests, perforates the cell membrane. So it destroys the cell membrane, which then means, obviously, the cell membrane's not selectively permeable. Everything can get in and out, and it all goes haywire. Bye-bye cell. Okay, so that's the first type. Then you've got TH cells, and the H stands for helper. Those ones are super important in that they basically help other parts of the immune system. So some of them stimulate phagocytosis. So they will be like, right, come on phagocytes, we need you to crack on and destroy any antigens, any pathogens that you can. And the other ones stimulate B lymphocytes. And then when we look at this next lesson, I'll explain how that works. And then the third type, T memory cells. These are the ones involved, these are the ones involved in immunity. So those are the ones which, next time you are presented with this pathogen, they will respond super quickly, and they will clone quickly, and they'll differentiate quickly, and they'll make the response happen so quickly that the pathogen can't make you ill because it's been destroyed super speedy quickly. Thank you. Goodbye.